Thank you everyone for coming to today's Brown Bag. So today we are featuring Dr. Brittany Pequegnat. She will be talking to us about the topic, Chemistry, a Sweet Experience. So we are speaking from Vancouver and acknowledge that we are doing so from the traditional unceded ancestral territories of the Musqueam, Squamish, and Tsleil-Waututh First Nations. Before we begin, please note that today's event will be video recorded, and we ask that you keep your phones on silent during this event. If you have any questions, please direct them to the very end where there will be a Q&A session. For more information on SQUIST events, please visit our website where we have events all throughout the year. Currently, they're online. One of the events that's happening, I think, next week is the Women in STEM Virtual Job Fair. Here we are featuring over 13 companies and 300 registrants are attending this event. We are featuring companies that are startups and well-established corporations and have on-the-spot hiring. So if you would like to join us, please do so and check out our website, squist.ca/events. So now I would like to introduce Dr. Brittany Pequegnat, and she is currently a tetanus antigen subject matter expert from Sanofi Pastuer. So Brittany, please take it away.
Thank you so much for the wonderful introduction. And thank you to everyone who is in attendance here today. So this talk is to go over my journey from academia into industry. And I thought a lot about what I would say today, mostly because I'm an overthinker and also because for those of you who know me, I don't normally talk about my own experience that much. But I came to the conclusion that today the real information about my career that I want to share with you is sort of the decisions and the lessons that I learned along the way. And I just want to preface my talk about the idea that each of us sets out on our own journey. No two journeys are going to be the same. And even if you're ending up in the exact same place as someone else, you probably took a completely different path. So as I talk about my journey today, it's not for a comparison or for you to think, is my pace good enough? Or, we often get caught in this idea that we're not moving fast enough. And I don't want this to be about that. So just keep in mind, I did this journey on my own to get where I am here. And each of you will do what you need to do to move your careers forward as well. So I started my journey in academia at the University of Guelph, and I chose a unique program. I chose Applied Pharmaceutical Chemistry in Technology. And the reason that I decided to go this route to go into university was that the program interested me in the sense that not only would I get unique cooperative education experience, but I got to spend 8 months at college. And I went to Seneca College in Toronto for 8 months, where I got to work with pharmaceutical industry experts and learn hands-on techniques for, pharmaceutical processing. And I would say, as I walked through my undergrad, the main lesson I learned at the very beginning was that you should never give up on yourself. And I would love to say that I entered university and I took chemistry and I excelled, but I didn't. I actually wanted to switch majors in my first year because I did so poorly in chemistry. And I went to my program counselor and I spoke to her just about what could I do next instead. And she sat me down and she said, the hardest part is always first year, starting and getting your feet on the ground. But it gets better if you stick it out. And she actually went through my grades with me and she pointed out something that wasn't even obvious to me at the time, but she said, you excel at lab work. And if you keep going, second, third, and fourth year, you'll have a lot more lab work and more opportunity to show who you are as a scientist. And I took that to heart and I stuck it out. And obviously, I stayed in chemistry and I ended up loving it, specifically analytical chemistry and organic chemistry. And I got to apply my academic knowledge to some co-op experiences. And in my first co-op experience, I was placed at McNeil Consumer Healthcare, and they're the makers of Tylenol. They're based in Guelph, Ontario. And I worked in their quality control department. And in this placement, I was exposed to new testing methods. I got to learn HPLC, dissolution technology, separation of drug products. But the key here was I learned two things about myself that would help shape my career moving forward. One, I was not cut out for QC work. I enjoyed learning the techniques. I enjoyed performing the experiments, but I preferred to have something more dynamic. And I found in QC, the repetitive nature of the tasks just wasn't a good fit for me. And because I realized this, it led me to the second key learning at this job experience. And that was, I need to be busy all the time. And from that, I actually learned the value of volunteering. I used my free time not only to volunteer on projects at work, such as learning the Lean and Six Sigma process and helping implement it for FDA audit, but I learned how to volunteer in the community as well. I helped out for Take Your Kid to Work Day. I helped out at science fairs. I even helped run the kids event at the company Christmas party. And I really learned that work doesn't have to be the whole balance, and I could volunteer my time as well in the community. So as I entered into my second co-op term, I opted to go back to McNeil. They asked me to come for a second term, and this time I went into the research and development department. And here I was now a scientific services employee. And this job was interesting because I got to service the analytical equipment. I learned all about HPLC, how to take it apart, how to put it back together. And I learned that from the technicians from Agilent. I also worked closely with Mettler and Toledo and with Thermo Fisher. And I really remembered the value of hands-on work and how much I loved kinetic learning. And lucky for some professors later in my academic career, I used those skills and my confidence to rebuild HPLC units at Seneca College. I serviced the HPLC units for the analytical lab at Guelph, and I even tackled taking apart our GCMS in my PhD lab when it went down, and I made them an SOP for it. And then my final co-op placement, I think this is the one that taught me the most about my career going forward. And I switched gears and I went into the food industry. And this was to challenge myself, make sure I wasn't biased into a love for just pharmacy, and to also go back to QC, I know, and take one more shot. And in this job, I learned all about dairy products. I learned about food safety. I did microbiology work. And because I love to volunteer time, I took on the big project on the factory floor, and I updated all the SOPs. And in order to do that, I took an interesting approach, and I shadowed each employee for a day. And then as I worked through rewriting the SOP, I actually worked each of the line jobs in the plant. After that, we went through the SOPs together, and we made sure that they were effective for new employees that would have to take over the jobs. But here, the value I really learned and that I still carry with me a lot is that you need to submerge yourself in all aspects of what you're doing. So at a company, you need to understand the end goal, and you need to understand each of the jobs and their importance in the end-to-end processing. And I really learned that by doing this. I worked closely with QC. I worked really closely with our QA professionals. And I also had to work every line job. And it was a very fulfilling experience for me. So after my final co-op placement, of course, I neared the end of my undergrad. And like most undergrads, I had no idea what I wanted to do. Did I want to graduate and go into industry? Did I want to try to write my PCATs and go to pharmacy school? Did I want to go to grad school? Did I want to become a doctor? And I was really anxious, and I got really lost and really confused. And I then just registered for courses in my final semester that maybe I wouldn't have chosen before. And one of these was bioorganic chemistry. And on the first day of this course, I learned that it was all about carbohydrates. And I learned that with carbohydrates comes the idea of puzzles and problem solving, because there's no databases. You have to build your own characterization. I fell in love, and the rest really is history for my career. I went to my co-op advisor and I asked him for a recommendation letter for grad school. And he ended up inviting me to his lab for the day for a tour. And he was a vaccine-based lab for carbohydrate chemists, and I never left. So I joined Dr. Mario Montero's lab for my master's, and I had the very unique opportunity of characterizing bacterial polysaccharides and then developing conjugate vaccine candidates. And I focused mainly on bacteria associated with gastrointestinal issues in autistic individuals. And I was able to actually characterize a repeating unit and develop a conjugate vaccine candidate. And we went on to publish my work, and our lab now holds patents for this technology. And in this process, because of the importance of vaccines and the idea around autism, of course, a lot of press came with this. We were interviewed by the school. We were interviewed on CBC News. And in this, my professor taught me more than just carbohydrate chemistry. He taught me about sort of the grace of having acknowledgement of your work. And he taught me a lot about why research matters. He was very humble towards helping others, and it was inspiring for me to see. And it really shaped who I became as a scientist, because he wasn't about, the flashy diseases and disorders. We were working on gastrointestinal problems, which people often don't like to think about. And because I wanted to help people and improve the quality of life, after my master's ended in his lab, I made the choice to continue there as a PhD student. I felt my work there wasn't done, and I really wanted to learn more and as much as I could about carbohydrates. But during this time, my professor was very clear with me. He was going to push me and he was going to challenge me. And he wanted me to be a well-rounded PhD candidate. So he gave me the opportunity to synthesize my own disaccharide molecule as an organic chemist. I learned ELISA work by going to work with another lab for a couple months at his request. And then he challenged me to grow my own bacteria for one of my PhD projects. And while all of these experiences were very challenging, it helped to shape me as a scientist and build my confidence in my abilities. And as I neared the end of my PhD, I discussed with him the ideas of industry and academia. And he told me, based on the challenges I had accepted, I was ready to do either. Throughout my master's and PhD, I also went on some conferences. And in this time, I did the important thing of networking myself. Based on these network connections that I had made, I was able to get interviews at a vaccine company in Montreal, at a glycan and drug development company in Hamilton, and then also at a stem cell and glyco-biology lab in Boston. And of the three, the job in Boston was the most outside of my comfort zone, and I knew it would be my biggest challenge. So when I was faced with the choice, I had to really believe in myself and really think, you deserve the opportunity that you're being presented with. And I decided the hardest career path, and I went with the support of my professor and family to the US and to Boston for my postdoctoral research. So I took a job as a postdoctoral fellow at the Brigham and Women's Hospital, and I had an appointment at Harvard Medical School. And it was a very challenging experience for me. I went in as a chemist with a strong analytical and organic background, and I was asked to learn cell biology, glyco-biology, immunology, all about stem cells, mammalian tissue culture, and it was very overwhelming. I read a lot of textbooks every night and a lot of publications every day. And if it wasn't for the wonderful people in my lab, I honestly don't know if I would have made it. I used my experiences from my undergrad, knowing that you can rely on the people around you to help you be successful, and that it's okay to ask for advice and help when you need it. And I allowed myself to start to excel in my postdoctoral position because of this. And during this time, I was also presented with a sort of collaboration with another lab that did glycochemistry. And in this, I got to use a little bit of my knowledge from my PhD. And it started to become clear to me that while this experience was great and I was learning new things I could apply later, I really missed chemistry and I really missed vaccine development. So I reached out to my PhD professor, again, pulling on those mentors from around me. And I talked to him a bit about what he thought and how I could maybe start to transition maybe to another academic lab. And he explained to me that departing from academia doesn't mean departing from learning. And I think that was a really key concept for me. And he told me, you know, there are some startups in Boston that maybe you can look at, and it's a good way to segue yourself into industry. So I went on some interviews, I gave some talks, and I was offered a position in one of the startup labs in Boston. And I started a position at Afinovax. And I got to work with some amazing scientists developing pneumococcal-based vaccines using their patented technology. And I was a science there. I led their new vaccine development group. And my boss and mentor, his name was Puvi, and he was one of the founders of the Pfizer Prevnar vaccine. And it was an amazing experience to learn from him and learn as much as I could about taking a vaccine to market and making sure it's successful. He also pushed me to learn everything I could in the lab. I learned every QC role there. I got exposure to scale up. I learned test method development. And I learned a lot about troubleshooting processes that had already been tech transferred. This job, I feel, was the perfect mix of academia and industry. And little did I know it at the time, but it was preparing me for the biggest jump I was about to make. And after a short period of time at Afinovax, I was faced with personal decisions, and I made the choice to move home to Toronto. And when I made this choice, I had to apply to jobs to make sure I had something to keep me busy when I came back. And I started to apply to jobs that maybe were a little out of my reach in things like experience as far as years. And I really looked to see, okay, do I meet 75% of the things these people are looking for? And I was actually selected for an interview at Sanofi for a position in their manufacturing technology team. And I had previously had contacts at Sanofi, and I reached out to them a little to find out about the team. And they told me this would be a good place and a good fit for me to interview. So I accepted the interview. I was very nervous because I didn't know a lot about tetanus. I worked with Clostridia a lot in my career, but it's not tetanus for sure. And I was about to interview to be their subject matter expert. And the interesting part about the interview process was that the managers in the interview helped me to understand the value of my previous experience and how I could apply that to the job that I was interviewing for. they pointed out things to me that I was being humble about. I had managed undergrads when I was a when I was a PhD candidate. And that's like managing employees in a company. And they pointed out that I had, looked at grant money before, and that was like thinking about budgeting. And when they started pointing out these similarities, I felt more and more comfortable in the idea that, yes, I had the experience and I could tackle this job. And really the biggest challenge that they saw for me, and that I also saw for myself, was I was going to a company with over 1,000 employees, and the largest company I had ever worked at had 25. So we talked it through, and I said I could overcome this and take on the challenge. And they hired me on as their tetanus subject matter expert. The first few months in that transition period were very overwhelming again. I moved home. I had to learn a whole new bacterial species. I had to reacquaint myself with GMP, and I had to meet so many people. And again, the value of a team and a strong community around you is what saved me in these first couple months. Everyone showed me, how to use things, where to go for help, who to talk to, how to get things done. And once I understood the system and started to get into the groove, it became very easy. And I learned that my experience that I had in my CoOp placements and my master's and my PhD and postdoc, they all set me up for this and for success. And I've been at Sanofi now for almost 2.5 years, and I really can't imagine a better transition for me into industry, just being in the vaccine-related field and having that experience and building it through. And I think with that, I just want to say this is the beginning of my journey, and I'm hoping that what I conveyed today and what I've experienced and my transition from academia to industry will just provide you with some insight on how you can do the same, or even insight on how someone got to the same place as where you did. And I think my take-home message that I really want to make today in my talk is you should ask for help when you need it. And I think mentorship and having a mentor to help you transition from academia into industry is something that you should look for and find value in, because each person, we're going to navigate challenges and we're going to have to find new opportunities. And these people are there to help you, and they want to see you succeed. And the main lesson that I've learned is that that's okay. We are all on our journey together, and we all have somewhere where we want to be. And I think that that's one of the most important lessons to take away from my experience, for sure. So with that, thank you again for everyone for coming today. And I think we'll open the floor for questions.
Wow, that was an amazing talk. Just because I finished undergrad like not too long ago, and I'm going into immunology. So I could never imagine myself going into chemistry because I'm absolutely terrible at it. So, so good for you for getting out of your comfort zone. And yeah, that's an amazing experience. So we'll start with our Q&A session now. We already have a question in the chat. It's how beneficial is postdoc experience to transition between academia and industry? Can you point out pros and cons?
Okay. I really think postdoc experience was important for me in the sense that when you're in your master's and your PhD, there's a lot of guidance from your supervisor. In my lab, there was for sure. And, once you transition to that postdoc experience, there's a little more onus on you to lead your projects, push yourself forward, find the resources that you need, find collaborators. And it just kind of sets you up for figuring out in an industry setting, that you need to be the one pushing forward. You can't go to your manager every time and say, who do I talk to next? What's my next step? It's okay at the beginning to get your feet wet, right? But then once you're in the system, you need to do it on your own. And I think that postdoc experience and the challenge that came along with finding resources myself and all of that, it really set me up for success in industry. As far as cons go, when I thought about doing my postdoc versus going into industry, the real consideration was I still was unsure if I wanted to try to go the professor route with my career. So I figured staying in a postdoctoral role, it would give me the opportunity to maybe publish a few more times or try to patent something else to set me up for success if I wanted to apply for an assistant professor position or something like that at a university or college. I think it's really up to you on if you think it's going to be beneficial for your pathway. But yeah, that's why I made my choice. That's all I can say.
And another lead on to that question is also you talked about your experience with vaccines in grad school, which I think definitely helped you with your transition. But I was wondering how a newbie to a biotech can make their path with less relevant experience.
I think you need to look at the strengths that you've acquired through your degree. I don't know specifically what you would be transitioning with behind you, but in each role, like for example, in my role that I took, I wasn't an expert in every single aspect of my job when I joined. I had had exposure to a lot of things, maybe once or twice in my academic career or an early industry career. But then you're able to pull from that to help you move into your next step. So if you have a little bit of immunology background, maybe you can apply that to the job that you're applying to, but you have to see how you can make that work for you, right? And I think the other thing we get stuck on is I was lucky enough to find a job that was very close to my interest, but sometimes your strengths can get you in the door in maybe a related role. And then you can work within your company and talk to them about how can you transition to something new and something that you want to learn. And that's another important part of transitioning. You don't need to enter into a job that you think is perfect for you, but maybe one that will help set you up to reach that dream job that you're thinking about.
Yeah, of course. That's a great point. I think sometimes when we look at a job description, we see how many things we can actually like check off. But and then let's say you check off all the boxes and then you go to the interview and they're like, oh, which has happened before. And people will say, well, now you don't have anything to learn. Like there's nothing more for you to upgrade because you already know everything, you know? And that's something that I've heard happens. It hasn't happened to me yet, but that's also something, you know. So it's always great to kind of be open to learning and all of that. Another question is also you talked about your experience with cell culture and new techniques you learned at Boston. I was wondering how you addressed it in your interview that you have no experience in that part, but can still make a great fit.
So for my job in Boston in the postdoctoral position, when I joined the lab, they were actually looking for someone with glyco-chemistry experience to join their group to help with a collaboration they had with a mass spec lab. And when I joined the team, that part was still fairly new. So I was sort of pushed more towards learning the cell culture aspect of the position. It wasn't necessarily the choice for me for the position itself, but it was my strong background in the chemistry component that actually helped me get my placement at that lab in Boston.
Okay, and another question is, can you describe your day-to-day in your current position at Sanofi?
Oh, so my day-to-day activities, well, right now, I have a lot of project support that I have taken on in my position as well. So a lot of my job right now is providing support to the manufacturing shop floor, so our production facility. I support from fermentation all the way down to the final sterile filtration of our product. I often attend a lot of meetings, but I'm consulted on any deviations or any investigations into the process. And I also provide protocols and reports. I help with trending of the process parameters and help with statistical reports. And then I also support any change controls or CAPAs that come through the system that are affecting the tetanus manufacturing process.
Wow, that's pretty interesting. That's a lot. Um, so a personal question that I have is sometimes when you're looking at new jobs or let's say a position in a lab, some often I get people say, you either have one of two choices, where one of the labs has a nice environment, a very welcoming environment, but it's not a very well-known lab. And future career prospects, it's not very great, I guess. I don't know. And the second option is usually a lab that's not very inviting, but it's very prestigious. And if you do very well, you're going to go far in your career. So, out of these two options, how important do you feel that that your work environment is when you're trying to decide which lab you go into or which job environment you go into?
I think work environment is very important. I won't go to details, but I have worked in both environments.
Mm-hmm.
I worked in lab where it was very inviting and maybe not as well known. And I worked in a lab very well known and maybe not as inviting. And while I gained a lot of experience from both, and both for sure helped my career move forward, I can tell you that the toll of working in a lab where you do not feel very invited and warm is a lot to take. And luckily my lab mates were always very nice and very inviting, but even, the pressure of producing quickly, or it overwhelms you to the point that you don't really even enjoy the experience for what it is. And I really think it's valuable that you enjoy what you're doing, because the more you enjoy what you do, the more you'll actually end up learning and challenging yourself. Even in a lab that's not as well known, you can do amazing research, and it's about then how you market yourself.
Yeah.
Right? You're going to carry whatever you take with you. And as long as you present yourself well and you have the background to really push yourself into the positions or another lab that you're looking for, it doesn't need to be a flashy, well-known lab for you to be successful.
Another question is, do you have any tips on how to learn new skills quickly on the job?
I wouldn't say quickly. I think the best way actually to learn new skills is to take your time and to learn them well, because then they stick with you longer. If you're trying to learn things quickly, though, you can always shadow people who are experts and sit down with them and try to understand. But I think really the best way is to experience the skills over and over on your own and really solidify them and take your time to really internalize what you're learning so that you can apply it in the future.
Okay, and for your career path, do you see yourself staying in a very technical SME role, or are you considering a project lead or program manager position in the future? Why would you not be interested in a project lead program management position?
So I would say right now I'm looking more at staying very technical in a more SME role, mainly because I don't feel personally I'm done developing that side of my own career. But turning into a project lead or program manager in the future is definitely something I have considered. I've also looked into things like taking, my MBA and learning something like that as well, just to sort of continue my learning and go back to school. But I think you just have to know if you've sort of bottomed out or topped out, if you want to say, in your technical role, if you're ready to go into leading a project or managing a program. And I would say too there, I've had experience now with dotted line management and learning how to manage people. And that's also very valuable, learning those leadership skills and being able to apply them later. So if you can get involved in a project and start to learn what it means to be a project lead and a program manager in the future, I would start there for your own career development. I wouldn't say there's a reason I'm not interested in transitioning to that. I think it's always great to learn something new because you can always go back to being a technical expert, right? You can stay current on what's going on and you can transition your way backwards.
Mm-hmm. And so is there anything that you miss from academia?
Of course. I miss just running, whatever experiments I want every day. No. Yeah, I miss lab work a lot. I mean, in my role now, I'm not as much in a lab. So of course you miss that, right? You've been in academia I was there in master's, PhD, postdoc, 9-10 years, right? So you miss that. You miss the hands-on work and getting to really sit down and maybe for a day just think about a topic and not have to be in the go of, keeping things moving in a company. You can take that time for yourself and really understand something and maybe try to apply something you've learned before to something new. And that's definitely something I miss for sure.
Yeah, right now I'm working at a biotech company, and so I'm usually in the lab, but my supervisor, who's a scientist, she's usually in her office, and so sometimes she'll just come in and stroll in the lab and just be like, "Hey, what are you doing?" And I'm like, "My experiment." She's like, "Do you want me to help you?" And I'm like, "No."
Yeah,
And then
I
you can
understand.
tell, yeah. So I can see, yeah,
Mm-hmm.
yeah. So.
Sure.
Yeah. Are there any other questions? If so, you can either unmute your mic and ask away, or you can type them in the chat and we'll definitely get to you. So I have a question.
Mm-hmm.
Do you feel like doing more exploratory work at any time?
Like going back to R&D?
Yeah.
Yeah, that's something that I do think about. I know it's nerdy, but I still try to read publications a lot.
Mm-hmm.
I still read a lot about what's current and up and coming in actually carbohydrate vaccines because I still find it very fascinating.
Mm-hmm.
And I actually recently started connecting with people on LinkedIn that are still carbohydrate chemists and learning about what they're doing. So I definitely like the idea of exploratory research and research and development, new product development. It's exciting, right? It's the phase one of any project. And I've worked the front end, and I've worked now the back end of keeping a process running. That's a legacy process. So you do kind of miss that front end work of the excitement of discovering something new.
Yeah. So do you still attend conferences while working with Sanofi?
I had intended to attend a conference right before COVID.
Oh, no.
But I recently attended the online version of the World Vaccine Conference. It's very interesting attending online conferences if you haven't done that yet. But most of the talks are pre-recorded, and you can watch them, and the speakers are there to answer questions in a chat, just like a Zoom meeting. So it's a little interesting. But I did attend. I watched a lot of the talks. It was very interesting just to hear about what's up and coming with, I guess, mainly COVID vaccines right now. But there are opportunities, yeah.
I was just gonna say, so do you think it's still important for you to continue making those networks at these conferences and things like that? Even though you're already in industry, do you think it's still important in keeping those networks alive? Or?
Absolutely. I think that you get to meet new people still. It's always nice just to meet people who are in the same field as you in general, because they can talk to you about experience they have, and you can often find connection points to help you maybe navigate something that you're faced with in your own job. And it keeps your network open, and you learn other companies that maybe you never knew about before. And not that you're looking to transition your career, but maybe, you can always have that in mind as that was a really cool place. And what I heard from these people seemed like a very good opportunity. And maybe down the road, that's something that you would look to do for your own career. So I do think it's important to keep networking open.
Right.
You never know what's going to happen in your life, if you're going to have to make a big move or something like that. So you don't want to close yourself off and not be open to making those connections still.
So we got some more questions in the chat. So what was the biggest challenge that you faced when you left academia?
I would say the biggest challenge going into even a startup was getting used to the rules of good manufacturing practices, that you can't just change whatever you want whenever you want. There are rules you have to follow. And I think that was really hard, right? Because in academia, often we think, oh, that didn't work, we'll just change this.
Yeah.
But if you want to do that in industry, you have to, file the right paperwork and talk to the right people. And that was a big transition for me. So I'm actually glad I had that little bit of startup in the middle. We were still designing and trying to get FDA approval. So there was still that little bit of, wiggle room to make changes before we were doing our submissions. So that was probably, though, the biggest challenge I faced was keeping to the rules.
Yeah. That's a challenge for me too. So what was the biggest challenge in Sanofi as a PPD SME?
Oh, so that definitely came from a Sanofi person. It's dealing with production. No, I'm kidding. The biggest challenge being an SME is that you're often involved in major investigations that need results ASAP, and everyone's relying on you to tell them the answer. And I think sometimes it's, it can be overwhelming, and you feel flustered, and you're just trying to figure it out to keep everything moving forward, because production can't wait. And I think sometimes that would be the biggest challenge of being the SME and someone that a lot of people are relying on for answers.
So another question is, how was your experience in the new world of pharmaceutical industry? Do you feel overwhelmed, and how did you overcome these challenges?
How was my experience in the new world of pharmaceutical industry? So just transitioning, I guess, into Big Pharma. Is that what the question is?
I think so.
So as I sort of touched on in my talk, I think the biggest transition to Big Pharma is the amount of people you're surrounded with and learning, navigating the different job functions and how to properly, get something done and all the forms and what order they need to be signed in and all those things. And I think the best way to overcome that challenge, or the way that I did for sure, is to talk to people who are in a similar position to you, to reach out and ask for help and get the understanding and accept that you're not an expert and you need the help. Because I think a lot of us, when you come out of a PhD or even a postdoc position, you've been an expert at something for so long, taking that step back and remembering, you're human and you have to learn something from the bottom again.
Yeah.
It can be a little overwhelming for you because you just want to be the best at something as fast as you can. So I would say, understand that there's always a learning curve and that you need to rely sometimes on other people to help you to learn something new again.
Mm-hmm. And another question is, I'm a biochemical engineer with a strong interest in postgrad education in applied immunology and vaccine development. What recommendations do you have for researching PhD labs or supervisors? As an engineer, I may be looking outside the engineering faculty and comparing all science/engineering faculty members at all Canadian and American universities, which can be overwhelming.
Wow. Okay. A lot of when I was researching labs was based sort of on what I was interested in. So I researched a drug development lab that was repurposing drugs that had gone through, phase 1 and 2 clinical trials and then were rejected, and they were repurposing them. And I thought that was fascinating. Did I have the background? Not really. But I read all their research papers and I started to understand, and I networked myself and I went and spoke to them and tried to understand what their lab was about. And I think something that I learned when looking into what labs I might want to go to for a postdoc or even your PhD is that you can reach out sometimes not to the supervisors, but maybe one of the grad students. Try to understand from them what the lab's like, what's it about, see if you have the right fit for their lab based on the technology they're using or what sort of education they had. And so you said you're applied immunology, biochemical engineer. Well, with biochemical engineering, you would still have, a background in, biochemistry, in chemistry, and those are all applicable to immunology and vaccines. I think you just have to look at what your strengths are from your degrees and how you can apply those to a new field, right? So even though I wasn't strong in immunology, I had taken immunology courses before. I had the general idea of human physiology from my undergrad, and you can start to build on what you know and start researching and really start to build, internally some confidence that you can be successful in a different field. Now, looking at Canadian versus American universities, yeah, it's very overwhelming. And I think the overwhelming part about going to the US is the transition of getting a visa, figuring out where you're going to live, all these things. And one thing that I can say about going to the US is no matter what lab you go to, someone else is going to be an immigrant. And it was a very interesting experience for me because I really connected a lot with my lab mates in the sense that we were all away from home. So if that's the part that, is maybe deterring you and overwhelming looking at American schools, I would say don't let that deter you for sure, because everyone's away from home and nobody knows anybody, and you'll all become really good friends because you have that in common.
So another question is, how do you identify mentors when you join a new organization?
Some companies will have sort of mentorship programs that you can look into, but you can also talk to your new manager or supervisor about if they have mentors within the company that can help you move your career forward. I think another thing I would suggest if you're trying to identify someone to mentor you is maybe to look outside of your own department and maybe find someone in a different department, just for a different outlook on the company itself and how to sort of develop you a little bit. But it is hard when you join a new organization. You won't really know anyone and you won't, know how everything works. But if you definitely talk to the management in your company, they should help you to identify if there are people there that can be a mentor for you. And sometimes you need to think about, do you want a mentor for your current role, or do you want a mentor for what you're aspiring for your career and where you want to move? Because those are two very different mentors to seek.
How important do you think mentorship is in terms of career development? Some people think that, they can do it all on their own and, they have all these great ideas, but how important do you think it is to have a good safety net and mentorship?
I think mentorship's very important. I mean, I enjoy being a mentor, and that's where I spend a lot of my volunteer time, is mentoring people. I think it's really great to have someone you can talk to, not just to have them tell you what to do, right? But to listen to you and to sometimes just give you that assurance that you're making a good choice, or,
Right.
Your choice is for you. So if you feel it's the best one, you should do it. Because sometimes we need that, right? We just need someone to say, you're not crazy, this is okay, right? And
Yeah.
I really think mentorship's important. And I've had the pleasure of having mentors in all of my roles, and I still talk to all of them.
Yeah.
Even though, I haven't worked with my PhD professor in years, we still email each other and sometimes get on phone calls just to talk things through and to see if things make sense and if I'm still on the right path.
Yeah, I feel like sometimes when you try to advance in your career, you have all these ideas, and having that reassurance from a mentor is one of the greatest feelings ever, because a lot of anxiety can be kind of released from that. So
Definitely.
yeah. All right, so we're hitting our last 5 minutes. So if you guys have any questions, you guys can ask away. So I have a question for you. So you worked at smaller biotech companies and a larger company. So do you feel like when it comes to like new ideas and new technologies being developed in, let's say, the R&D section, do you think that it takes longer for those ideas to get through, or do you think that having a longer process might be more beneficial because you're going through all of these like checkpoints that yes, this is a great technology?
Yeah, something I noticed working in a startup is often you're on a race against the clock because of your grant money.
Mm-hmm.
And we did have some pitfalls, of course, in what we did. You know, you push something through, we hit at scale, and we had a lot of issues. You roll everything back. And I mean, I know that happens in big companies as well, but I feel in smaller companies, because you're really trying to make that push, you might push things through a little faster than you should, right? And not that it's wrong or or not doing it the right way, but you do have that pressure of we need to perform for our grant. And
Right.
yeah, sometimes dragging things out and taking a long time is better because you shake things out earlier in the process and you don't have these bigger issues later on.
Yeah. Sometimes I'm in an R&D section, so we have these great ideas, and we're like, oh, it's going to be the best thing ever, and then we have to go through all of this long process, and so when I think about it, I'm like, we could have just done it, but it's like you have to go through the right process, and it makes sense. It makes sense, and it actually saves us a lot of money, and which is very important.
Yes.
So, yeah. Another question is, what types of processes or exercises do you use for scaling up processes in your current position? For example, FMEA, failure modes and effects analysis.
So we often start off by doing small scale or feasibility trials. We have a lab that we can do that in on site. And then from there, we can look at, what the output of our small scale was and, learning how to then apply what we learned from small scale into larger scale models. So yeah, you could use things like FMEA. If you have, pitfalls in your small scale, you can look at, the 6 failure modes or something like that as well. But I think the main thing is, these small scale trials, understanding if it's scalable, if you can scale it up. We also use engineering runs. So you're going to be at scale, doing a run in the production floor. And that will give you more insight into whether or not something's going to work, looking if anything doesn't conform to how you think it's going to work, going back, doing a rework, figuring out how to move the process forward. Those are all very important things in the scale-up process.
All right. Well, thank you so much, Dr. Brittany, for coming today and answering our questions and for having that amazing talk. I feel like everyone can relate to bits and pieces of that. And from the chat, it seems this is an amazing talk. So I'll let you guys go. Thank you so much for everyone joining with us today, and look out for our next brown bag session on the swiss.ca/events website.